clear off the deck
Do an
still?
intro, Sylvia.
Well, all right. Yes. Yeah.
1 meter off.
That would be great.
Coming down. Coming due. And 2 new skins.
Mm-hmm.
Let them know the camera's not working too.
Oh yeah. Hi everyone. My name is Sylvia Knupp. I'm from Montana State University. I'm directing here today. I'm joined by Emily Aguilar-Pine from University of Texas at Austin. One's been fired. We're
Two's
here.
been fired. Nice.
Right.
Camera working.
Nice.
We're about to take some cores on the seafloor here. We're off the coast of Uruguay on the slope of the continental shelf in the Atlantic Ocean. So we're going to take some samples from the marine sediments here, looking for Asgard Archaea.
We just want to get a little clearing somewhere.
As you can see, we've got a lot of starfish on the bottom here. We're looking for a good place to take sediment without disturbing a ton of them.
Don't get too close to it either because we've got a comfortable porch.
We're missing one of our cameras today on this dive, so we're operating on the other cameras to try to core here. Oh good, you can see those.
Like, what's your preference for ones to go first?
I think the Geochem should go first, and
Special
so
ones on the
Yep.
on the porch.
on the porch. If it would be possible to make sure that 9 and 10 can sit next to the rest of the cores that we take, like 1 through 5.
Yeah.
That would be great.
Right. Right, so you want it only next to 1 and 5, or do you want us to do 6, 7, 8 as well? 'Cause they're going to be easier to get them out the way first,
Yeah.
and then access the back ones. Sure.
Yeah, let's do that. Because I think we'll just take all of them that we can.
I just have an extra sample sheet that we can use. We're not full porch out there. That's great. We have a little more out. That's really sick.
Actually, I'm better using the other.
Oh, no. One with the drones.
Porch line? Porch line for them?
Sorry.
They're looking for a porch
Like,
plan
if
so
we
they can
come
know.
over-
Oh,
Yeah,
yeah,
porch.
porch plan.
They're coming out.
Do we have a porch plan?
I've got
No,
one.
I did not make one up.
Pull out.
Jules, do you know what it is?
I can
tell you
draw
where-
it. I
I
can
can
draw
tell you
it.
where they're in the folder.
Is it in the folder?
ROV shared drive.
Are
Then-
we sure? I don't have that. Hey, don't worry, I'll put
Ask-
the push core in it.
Ask Chris.
Yeah.
And then it's in this booze inside the ROV shared drive.
Alright, let's go use something better.
And is that you fully porched out?
That's full porched out, Crouch. Confirmed. Let's do.
Yo.
Just
to let you know, we don't have the science, what we call the SciCam, which is a science cam we use normally to
So you guys want
give you all a better view of what's around us. That hasn't been working since the surface is blinking in and out, and now it's gone. So I'm not going to have a whole lot of good views of things, but we're going to get
on the
our
Geochem
samples
All the Geochem ones, and then 6 and 7.
Oh,
can
yeah.
go
at
down
the
to-
station
Yeah, all of them.
that we were permitted to.
Yes, perfect. Let's do HD3.
Are you done?
Actually, I got you.
You got it?
Yeah. is 6.
6, yeah, 6.
You want
So,
that one there, Mike?
Do we try and scatter? Try and scatter these guys, or do we just go for it?
Just try and work around them, yeah.
Mm-hmm.
I don't know.
They're on the same location, just about. Mm-hmm.
Like that view, or do you want?
Yeah, that's better actually. Yes.
And then I can switch it to what I get for better view and push it down.
I'm gonna just try this one here.
I don't. These ones are not kind of scoopy ones.
Push for
Yeah.
6 in the ground. Yeah, we're at the same station that we were yesterday, as the sediment looked really good for our microbes. It was highly sulfidic, so probably, and we measured oxygen as well, and oxygen was fairly low in the sediment profile. So currently a lot of our organisms that we know about, these Asgards that we're looking for, many of them are anoxic, so grow without oxygen. So this looks like a good place to sample for them and try to find them. Push core 7. Down.
Thank you.
These guys need to move.
Attempting to sample around these sea stars, but the ground is kind of covered.
Oh yeah, there are a lot.
They'll move away.
Like going down here.
Better.
Enormous.
Perfect. So, looks like we're about 900 meters below sea level here. We're on the slope of the continental shelf, so getting a little bit deeper samples today.
For those of you that are regular viewers, I'm thinking of Lisa. We will be diving. We should have the camera fixed tomorrow. We'll be diving to 2,600 meters on the continental margin. Should be exciting.
It's number 8. Push core number 8.
Beauty.
You were saying 1 through 5. I don't think we're going to manage 1 through 5
Okay.
for this, because it's on the opposite side
Mhm.
where we're going to go. It's probably going to be the least likely ones we're going to have success with.
Okay. Is it going to be possible to get the ones on the other arm?
Yeah, I think so. We'll try those ones for sure.
Okay. I think we can put all of them down in this location.
Sure. Yeah. I think we only need 5 in addition to the.
5 additional ones.
Do you want to try and like put these ones in? I would suggest putting these ones in and putting them back and getting the porch out the way,
Yeah.
Okay.
and then try
Yeah.
the other ones just to see how much success we get with
Yep.
Perfect.
Sounds good.
using these.
Yep.
Mm-hmm. Yeah. On my side of things, I'm looking at trying to culture organisms from here. So bring them into the lab and grow them and close to their natural conditions, but in a way that we can study them without coming back to the field. So I'm really interested in getting as much of this mud as possible.
Sylvia,
to
this
come up.
is
Mhm.
9.
Oh yes, this is 9, push core 9.
Thank you.
Looks like we scared a lot of the fish away as well. This is push core number 10. Going in right now. Along with trying to culture these samples and sequence their genomes. We also are doing some geochemistry of the site, measuring oxygen, pH, sulfide levels, potentially methane levels, to try to get an understanding of this very specific location that we're at. So that's what these cores going down are. Push core 11.
Try and create a spot here.
Ideally next to one that is not geochem or like next to the not as special ones, like for example 7 or so.
Not next to 7.
Next to 7 will
Next
be
to
good.
7.
Yeah. Not next to 8. Next to 7 will be great.
Try like this little patch here.
Yeah,
put
that will
these
be
guys
great.
out there.
Yeah.
Good.
Just lethargic. There you go.
Perfect. If there's a little bit of water left in the headspace of this one, that will be ideal.
So how
Hard to see
there.
from.
Mm-hmm.
Yeah.
I think that's good, yeah. Mm-hmm.
So I could give you a zoom on this camera if you want a screenshot.
Yes.
Perfect.
Yeah, we just want the numbers.
Maybe the numbers. green one's 6 anyway.
Yeah,
Is it? Oh yeah. it's only been 6. I'm just right.
What's the red thing?
Sure.
Shrimp. Little shrimp.
That's his formal name.
Is it you?
Yeah.
Little white spots.
You guys get there, that one?
Did you get it?
Yep.
Yep. You got it.
Just
Perfect.
got it. Thank you.
So.
We have a request for an instant bottle in this site.
We got 2 already.
Wow, okay.
Do they need 100% of both of them? Do you know?
I have no idea.
What?
Do you know if they need
Maybe zoom in a
100%
touch on
of
the
both of
ascent.
the?
I think they
For
just
these
wanted
bits.
one,
Okay.
so.
Perfect.
Yeah.
Let's see how solid this sediment is.
Gracias a todos los que siguen escuchando y estando al pendiente de la transmisión. Aquí vemos los ofiurios, estas estrellas de mar. Vamos a ver cómo se ven estos sedimentos. It does look.
That's 11 coming back out.
Yeah, they look good.
Nice.
Looks good.
Yes.
Yeah, awesome.
Perfect. I'm also glad that they move away. Would rather not sample
It's
them.
in Microbial shared drive, cruises, made there. And then it's just that.
That darker mud at the bottom of the core seemed to be the sediment that we found to be most sulfidic. So that looks like a good push for it.
Yeah, thank you.
Perfect. Mhm.
You know. It's actually double it.
One sec.
Volume starts.
Okay, and I will zoom on this one. Yeah,
I believe
10.
so. Nice. Uh-oh, got one.
It's okay. We can just put it back.
Sounds good. along with trying to culture some Asgard Archaea, I am particularly interested in culturing methanogenic archaea, so organisms that produce methane from their metabolism. And a very convenient aspect of this cruise is that the same sediments that we're likely to find some of these Asgard Archaea are also ones that host my methanogenic archaea. So these samples are looking good for both for finding Asgard Archaea and for finding methanogens.
This time? Maybe? No, I think so. It should go through the system. We got another little nudge. This one's gonna be the trickiest one.
Aquí estamos viendo el mapa de donde nos encontramos a 918 metros de profundidad, recolectando estos sedimentos que eran bastante con olor a azufre, con olor a huevo podrido. Sylvia, what do you think about the sediments from yesterday?
They were my favorite so far. Very, very sulfidic, very low oxygen.
Very stinky.
Yes. Not as much clay as we've been getting from some of these sites, which make them more difficult to work with, but I think more likely to host the organisms I'm looking for.
Yeah.
Yeah. Particularly looking at the bottom of these cores, the darker sediment there looks the most interesting to me. That's push 9.
Yeah.
Push 9. Mhm. Yeah.
Ideally, so we have 6, right?
Mm-hmm.
Ideally 2 more, like next to them, and that would be great.
Yes. I'm gonna try to get as many as possible.
This one's gonna be boring.
Oh no. Because of the camera issues today, we probably won't be able to get a few of the cores on the one arm, but we should still be able to get a good number today.
Ahora estamos recuperando este push core, pero el sedimento se ve bastante bien. Seguimos aquí en Mission Control. Silvia está hablando. También aquí está Emily Aguilar Pine de la Universidad de Texas en Austin, tomando notas de todo lo que estamos viendo. Emily. Emily habla muy bien español, pero le da... Es muy bien. So, Sylvia, this is our second expedition together.
Yes, it is. We're both still processing sediment.
Processing sediments in the cold room.
In the cold room. All the time. The sediments need to be kept at 4 degrees ideally, because as we heat them up, we might disturb the microbial community away from what it is when we're sampling here. So we've got a cold room on board, and we do all of the sampling and everything in the cold room. As soon as the cores come back on shore, that's where they stay for the most part. It's more fitting to the environment I'm coming from up in Montana this time of year. So I'm a little more used to it, but the warmth here in Uruguay has been very, very nice as well.
Ahora estamos tratando de ser un poco silenciosos para que los pilotos puedan discutir la recuperación de este push core. Ahora vamos a seguir con el siguiente.
How many samples are we trying to get today? As many as we can. We're quite excited about this site. That's push core number 8. Yeah. The pilots are amazing. They're able to get these cores in so many different conditions.
I can really one eye open again.
Hey, but it's still on the basket.
Still in the basket. We might be able to get it back, but that one in this corner closest to the arm is the hardest one to get.
Yeah.
Because it's so close, you don't get the height.
Mm-hmm.
And obviously, you've only got this camera, you can't really tell where the height is. We almost got it, but almost.
Oh, come on. Come on now. My thoughts is that maybe we can do a video of our own movement and I can just watch it.
Les invitamos a todos a hacer preguntas. Estamos tratando de contestar, de responder a todas sus inquietudes mientras hacemos el registro de datos. Mando muchos saludos a quienes me están escuchando, a mi mamá, Marilu, a mis amigos de la Facultad de Ciencias de la UNAM en México, Rigel, un saludo muy grande.
Look at the gray sediment in this one.
Yeah, they seem to be having two clear, defined layers.
Mm-hmm. Good. That's going Mhm.
Looks like it could be right there.
Yeah.
This is push score number 7 coming up. Great. Mhm.
Now, this is the one. We're trying to put 6 in where number 9 was.
6 isn't special.
But it's going to be easier to get 9 in if it's at the front rather than at the back. So it's going to be harder to do it at the back because it's hard. Yeah. What do we think? What's the reward? I'll try and put it in. If it looks like it's going sideways, I'll just put it in the 6. Yeah, please.
Push score number 6. An interesting difference between these sediments and the ones that Val and I worked on our previous cruise is that these are all right at sea temperature. They're at 4 degrees Celsius. So it'll be interesting to see how quickly any archaea in the sediments grow up. As you know, a lot of other animals move more slowly, grow more slowly at lower temperatures. So this might be the case for
Yeah.
these organisms as well,
That's bad.
in which case we'll have to spend quite a bit of time waiting and seeing how they behave.
How long it took you to grow your methanogenic archaea from hydrothermal vent systems, Sylvia?
It varies a lot. So some of the hydrothermal organisms, once they, especially the archaea, once they get going, they're very quick. They're basically completing growth curves in like 7 days to 14 days. In contrast, some of these colder organisms will probably take, could take upwards of a month or 2 months, but it's hard to predict how fast an organism is going to be. So we have some from hot springs that are also taking months and months to grow up. So yeah, it's kind of it can be a little bit of a waiting game sometimes. You just give them the substrates and continually monitor them, and hopefully eventually they get growing. It's a little bit faster to get the sequencing results in, hopefully.
Yeah, you have to have a lot of patience, right, to grow these microbes. And a lot of waiting.
Mm-hmm. Yeah, but we can't really do it without the sequencing data.
Yeah.
we use the sequencing data to figure out what we're even going to target for cultivation, as well as with some deeper sequencing to figure out what the microbes might be able to do. We can't recreate the environment here perfectly, so we kind of use that genetic profile to figure out what, what chemicals they might like, or that sort of thing, what they eat, what they breathe, to try to recreate the conditions in lab. The geochemistry data that we're collecting also assists with that.
Nice. You need to do a side look for me and see if it even starts to slide itself.
What do we think? We'll want these other ones like real close. Can we come up, try and get this push core moved and come back down where we are? Or what do you think?
Sorry?
I don't know.
Can we move slightly and come back down?
Can we try to move a little bit so
Yeah, I mean,
This is
ideally,
easier.
if there is 2 more cores in that side, then we can.
Yeah, we'll probably be doing it off this other arm next,
Okay.
but I'm wanting to try and get this core back.
Back. Okay.
But we can come exactly back to this spot, but just
Yeah.
come up,
A little
move
bit,
a little
and-
bit, and
yeah.
then try and come back down.
Okay.
You happy with that?
Yeah.
Yeah. Yeah,
So we're allowed to.
just in case we don't find where exactly we are.
Aquí estamos agregando la posición de donde se encuentra el submarino para poder recuperar ese push core que está en la canasta.
Float and just try and keep that holes in view, because we want to try and land back.
Hablando con
and get the
Mike
other cores, like,
acerca de
next to
la
those ones,
estrategia a
and
seguir.
try and not stir up.
Come up and back.
Un poco de lo que estaba hablando Silvia es, nos recuerda qué tan complicado es recrear las condiciones del ambiente, las condiciones en las que se encuentran los microorganismos, para poderlos cultivar y poderlos observar y poderlos crecer en condiciones similares a las de su ambiente. Sin embargo, para organismos que viven a estas condiciones de alta presión o condiciones de alta temperatura, como la expedición que realizamos previamente, es muy difícil tener esas condiciones en el laboratorio. Por eso tomamos en cuenta la información del genoma para predecir cuáles son las posibles funciones de un microorganismo, y en función de eso decir qué nutrientes podría utilizar o qué fuentes de comida podría utilizar. Entonces, si es en microbiología se conoce esto como la anomalía de recuento de placas. Se sabe que hay muchos microorganismos, pero hay muy pocos que se pueden cultivar en condiciones de laboratorio. Por eso, secuenciación y el desarrollo de tecnologías computacionales ha permitido un mejor entendimiento de los microorganismos que no se han podido cultivar en el laboratorio y que sabemos que existen ahí, y eso nos ha permitido incluso descubrir nuevos linajes de la rama de la vida, como son las Asgard Archaeas, que se descubrieron hace 10 años por medio de métodos de secuenciación, y 5 años después se pudieron cultivar, pero justo la información del genoma y la información de las capacidades metabólicas predichas a partir del genoma fueron cruciales para poder determinar las mejores condiciones de cultivo.
Yeah, do some more porch work there, Mike.
Ahora
post up the map and see if you can see it.
estamos haciendo una maniobra para poder recuperar ese core que está en esa canasta y ahora Mike va a hacer tratar de recuperarlo. Así que estamos tratando de guardar silencio para no interrumpirlo.
This is our oxygen profile core that they're attempting to get back in the quiver.
Is that the geochemistry, Sylvia?
Oh no, you're right. It's number 9. It's the geochemistry.
Yeah.
Yeah. Number 9. It's the geochemistry that has the pore water measurements and I believe also the methane measurements.
Can you talk a little bit about the geochemistry measurements that you're taking with Yemen and like what kind of in a very general way, how are you treating the sediment core and what are the results that you're getting?
Yeah. So I'm working with Schmidt from University of California at Santa Barbara. So she's taking these geochemistry samples. If you see that tape on the side of the core, that is covering up holes that are drilled into the side of the core. The idea is to get pore water measurements for all of this geochemistry so that we can get a profile all the way down the core. So on this one, we're aiming to do sulfide, sulfate, methane, pH, and organic nutrients in the sediment. To get a whole idea of what the sediment profile looks like all the way down.
What is pore water?
Oh yes, pore water is water extracted from in between the sediment particles. So a lot of these measurements we can't easily do on the sediment itself. So, and we're kind of interested in the water flow through the sediment as well. So that's why we're profiling that. All of these measurements are pretty time sensitive because when things like sulfide interact with water or interact with oxygen, it gets reduced and that changes our measurement. So pretty much as soon as these cores come back to the surface, we try to collect as much of that pore water as possible and then measure these chemicals.
And why we're interested in having these measurements at a particular depth?
So something that we've been seeing for a lot of these sediments is that as you go deeper, the chemistry of the sediment changes pretty dramatically once you're no longer on the surface of the seafloor. So right at the surface, we tend to have high levels of oxygen. Oxygen still gets down into the sediment, but very quickly after you're above the top, sometimes top 2 centimeters, the oxygen levels drop off. That's normally when sulfide levels rise up. Sulfide doesn't exist very easily with oxygen present because they reduce, and so when we're interested in organisms that need a high level of sulfide or are fairly anoxic and can't tolerate high levels of oxygen, we look for sulfide and then sometimes sulfate in the sediment there. Methane is going to be very nice to correlate to try to look for methanogens, because if we're seeing methane in the, oh, there we go. You can see the sea stars on the bottom. If you're seeing methane, then it could be from organisms producing that methane. We're not likely to see high levels near the surface of the sediment. We're more likely to see higher levels down the bottom of the core, but we don't know. For these sediments, we don't know if there's methane production at all or at what levels.
Wait until the seabed clears a little bit.
And of course, there's a high level of organic material at this site. You can see the marine snow, the particles floating down to the bottom. So we're interested in the
Maybe it's better just to
organic profile as well through the sediment. And it may be that some organisms degrade them all at the surface, or it might be that some of that organic matter trickles down deeper into the sediment core, and we find organisms consuming that leftover organic matter near the bottom of the core as well. So all of these different measurements together are going to give us a better picture of where our organisms of interest are and the overall geology of this location, at least in the depths that we're able to measure here.
Right now we're just waiting a little bit to get to have a better visibility here before we proceed with the next push course.
Yeah, we were sitting at 12 meters before.
Bodes well for the amount of organic matter and stuff at the seafloor when it's really light and fluffy like this, but it also makes it way more difficult to sample. Well, at this level it's clear.
Away from our site.
Yeah, we're gonna let Liz clear that spot, and we will go back and find it.
Vamos a esperar un poco hasta que se mejore un poco la visibilidad y después regresar al punto donde Mike y Jess lo marcaron para saber exactamente las coordenadas y después regresar a muestrear ahí. Seguimos al pendiente de sus comentarios y de sus preguntas. Un poco recapitulando lo que comentaba Silvia, es el trabajo que se realiza para poder tomar medidas tal cual conforme el sedimento lo recuperamos del submarino. Lo procesamos en el cuarto frío, pasamos horas ahí procesando los sedimentos para poder disminuir un poco el metabolismo de estos microorganismos. pero también para obtener estas medidas que nos indican de manera indirecta la presencia o la actividad de estos microorganismos. Es como si dejaran una huella química de su actividad metabólica, y esa huella química es la que nosotros leemos para poder tomar medidas de esa actividad. Es decir, si hay presencia de organismos metanogénicos, si hay presencia de organismos que realizan sulfato-reducción. Ahora, bueno, vamos a tratar de mover un poco ese push core, a ver si puede salir.
This one is probably best to get.
At least we've got core 9 on the basket, but when we come back up, when it's positioned like that, it might lose all of its sediment. The pilot's doing an amazing job of manipulating what you can. Cruise science ends up being a little different than what we do at our home labs, because we have to work with what we're able to get. So with sampling, sometimes things go wrong, or sometimes we don't have the exact right equipment on board.
It's just not. I think it's wedged in there nice, but
I think that's
yeah, I think we're gonna have to give up on that one.
Yeah.
okay.
Porch. It's just right in a blind spot and
Yeah,
really good
that's-
place to try and get the arm.
That's fine. We still have number 10.
Yep. We still have another geochem
Yeah.
core. It'll be okay.
Do we want to take the sample onto that side? Do you guys want another sample of this push core too once we get back to this site, or?
Yeah. Yep, that would be great.
Okay.
just doing a language course on ascent. Do you think 2 more and then do the rest somewhere else, or should we try to just get what we can?
I mean, ideally, we just need 2 or 3 more since we lost that one. We don't need a lot of money in terms of. I think 5 less than
Let's try and get back to where we were then, Jess. See if we can just blow up and
Like a little drum.
see if we can get back to that same spot.
Roger.
Just nice and easy. Taking that port veil and probably torch as well. Torch going out with
No, they got started around.
Right, so if you just go straight ahead, just keep a viz on the see then we'll hopefully come across the bit good dead ahead from where you are, we'll come across the bit where we're at.
Sorry.
Still a little more disturbed here, but not as much as it was before.
Come down, settle down here and just have a closer look.
Yeah.
Maybe a little far across.
Try and zoom and see if you can see any obvious holes. It looks like a dark patch right in front of us. I don't know if that was. It's brittle starts to
I'm trying to re-find the place where we took the other course. Although with the sediment upturned, it's a little difficult to identify.
I think that's it.
Yep.
Think so? Looks like a few holes there.
Crazy looking, isn't it? Yeah, absolutely.
Thank you, sir.
Thank you, sir.
We're happy that this is the same spot.
Yeah.
Yep.
Yeah?
Yep.
Yeah. Let's see what we can do. It's the same. No, back off. Yeah, we need to change that. This guy. These ones are good.
The sea stars have already moved into where they were before.
So much worse.
It's there.
Really?
You can see the core's a lot better where that was. It's there. It's a bit blown out, but
Yeah, those are the outers.
the actual cores you can see the arm and see as well. What's going on there?
Oh,
Is that a thruster?
yeah.
Yeah, it's just a regular.
Yeah.
Okay.
Black smoker.
Alright, let's try to do it.
It's like Elven cameras that look. Shit. There's one. They have one really good camera okay on one of the arms. That is sort of like the Elven. Like there's the pilot. It's two scientists. And they're doing all the cams.
There's
The scientists are good. So it's just like you're like trying to figure out how to do it. Very like get good.
You want to turn on your uppers and turn off your hours. Yours on.
Do they have any idea what's wrong with it?
I don't know. Basically.
The visibility is quite low.
I think ours are about that. I'm gonna give it a little more than.
Oh, sorry, what was that? What number score is going in? I
In the
think
basket?
Oh, on the side here.
It's 12.
Push score 12.
I have a little bit of gold, so it's fine. So you can go past some stuff.
So bad.
That's looking a bit better.
Where do we want to try and put this? Just wherever's easy. Try to
Yes.
clear some.
Yep. Anything close to the holes already
Close
made.
to that first one. These little stars.
Mhm.
Not shifting.
They were moving so quickly before.
Just gonna try to go this down. How much are we realistically hoping to get?
If we
The further
could,
back these rows we get, the harder
Yeah.
they are
the
to
harder
get.
it is.
Yeah.
Yeah.
Well, you got. Well,
I the ideal number would be another 5.
Another.
Yeah, but if we can get 3, we
That'll
can
be
make that
great.
work.
Yeah.
Or
The minimum that we can work with is 3.
Mm-hmm.
So
do what you can.
yeah.
Yeah.
If that is unrealistic, that it's okay. We can work with what we.
We'll try and get as much as we can. This is
Yep.
coming here and then,
Mhm.
like I say,
This is push core 13.
Regresamos al sitio de muestreo y estábamos decidiendo cuántos cores son los que necesitamos, el mínimo número de cores que necesitamos para poder trabajar y hacer los diferentes análisis en estos sedimentos.
This is push core 14. You can only do so much. We try very hard not to disturb what we're not here to sample for, so if we can get the sea stars to move out of the way, that is ideal. Should be push core number 15.
We can get 2 as well, but do you want me to just keep
Oh
going?
yes.
Or want me to try and just call it 5 or what?
I think we can call it at 5.
Yeah, so we can do 2
Yep.
on the front as well.
Mhm.
So let me try and get one of these back in and see exactly what this is.
You want to compare the minutes?
This is push core 15 coming back out.
Ahora nos quedamos en el misión control, Emily, Silvia y yo.
Yes. Push core 15. Great. That's awesome. is number 14. Shore 14.
It's like you had all the cameras going with that one, huh? It's smooth, it's so
Yep.
number for
Fish core 13 being retrieved. These are really great cores that are holding onto the sediment well, so we'll have a lot to analyze back on the boat. And we have a fish observer, it appears. Push core number 12 being retrieved. The whole sediment kind of shakes. Just to clean the core out. It's probably very loose and silty. That can be pretty good for the pore water analysis, because if there's a lot more water in the sediment, then we have more to do analysis on. That's always good.
Ahora recuperamos más sedimentos y como recuerdan de algunas otras expediciones anteriores, el Mar de Furious. Muchas gracias a Jess y a Mike.
We're going all right. We could try and get the other ones if you want.
Yeah.
They weren't so bad.
Which ones?
We could try and get the back four if you like. I don't think they were that bad. They are
Okay.
going to be more difficult, but at
Okay.
least we've got
If
those
it's
ones.
not that difficult, then I would say go for
I
it.
don't say that. Chris is going to say it. Yeah, I said it's but
If we could get at least 2 more.
How many did we
try
get?
a
We
couple of
got
these. I think
4.
These back ones are going to be a little
Harder.
tricky to get, but these two maybe.
Yeah.
Try and
Yeah,
just
try
put them in.
try. We can try one and
And
see.
then we'll definitely get 2 on the porch. Anyway.
Yeah.
But
Okay.
Maybe 18 and 19 might be a little bit
A little bit more.
too tricky.
Okay.
I think that 2 more would be.
2 more will
very
be. Yeah.
Mike's just rock star. Like, you know
Yeah.
what? I know this. Give me 5 more.
Hey, that's great news for us.
So we don't replace them.
Oh, I'm done.
That's good.
Where are you going?
What core number is is this? It's it should be core number 16.
They're in order, yeah. It should be 16.
He's even at the coffee cup. So, we're gonna get
I'm gonna try and get a desk
right
too.
now.
I think the back one's maybe a little bit of an ask, so I'll just leave that. But we'll have got 2 on the porch, I number 2.
You couldn't get it out of there?
It's just, I was like, because I couldn't really see it on any of the other cameras, it was just being blind and I was risking of smashing up all those actual push doors.
You guys made it on number 6 and 7, and then number 2, which is this D4. And that will just be D4. Yeah. And then for the starboard one, we found the mids and the outers, so
This is push core 17. I'm impressed at how fast the stars move.
Yeah, they're pretty sensitive.
Yes. Good.
everybody see that.
So I put all the fragments on the course and see that
Except that darker gray sediment that I'm particularly interested in.
This is actually a better, like having that outer light is actually saved us. I don't think we would have got it with that light.
I think you did a great job regardless.
That's fast that time.
Hey, we got somewhere to go.
It's awesome. Even with the camera issues, we're gonna get everything that we need for today.
Actually, I should.
Wow.
It should.
Yeah.
Beautiful.
Yeah. Awesome.
Oops. What's that? Yeah, I think the back 2 will maybe be tricky. We can get 2 on that one on the porch that
Yes.
you want, just for an extra.
Yeah, number 2. And then
Yeah.
I think we're good.
Yeah.
Watch out for it.
Oh yeah. Oops. Whoops. Sorry.
Can you put HD4 on that?
Yep.
Yeah, it just gives you a nice little height.
What's that? Sorry. I didn't realize you were doing that.
No, you're not right. Sweet.
Hubo unos problemas con la cámara, entonces tuvimos dificultades para mostrar en este sitio, pero aún así Mike, Jess y Chris son unos pilotos increíbles, entonces vamos a poder vamos a poder recuperar bastantes sedimentos en este sitio. Vamos a hacer el número 2 y con eso terminaríamos el muestreo.
This is
Where
push
do you
core
want this
number
one?
2.
Put this one in this little patch here.
Yeah,
Here.
that's perfect.
Yeah, I'll just do that.
Zoom in. Might be go upward first. Might exit you out of average.
La característica de estos sedimentos, cuando los estábamos procesando ayer, como pueden ver, una capa negra al fondo.
It's too late now.
Esta capa negra nos indica la presencia de sulfatorreducción, lo que produce el olor a huevo podrido, que es el azufre reducido. Y bueno, procesar estos cores en el cuarto frío, uno debe de tener cuidado porque a veces estos olores se concentran.
We're gonna have to wait at the bottom for a little minute anyway.
Yeah, okay.
Because, well, depends. We've got slow descent before we really over the deck.
Oh, yes.
So whether or not you want to just explore this area a little minute and then
Yeah,
come
Let's
up
do
to
that.
give everyone a time to
yeah,
eat something and then
Yeah.
me and Jess will be
yeah, we will process them anyways after.
Yeah, so let's see if we can go around a little bit and look at some places.
You're too late. I saw you out there.
Oh.
I had the camera on you. We knew you were waiting for that last one, and then you're like, "Ah, it's good." Right. What's this?
Great job,
Thank
thanks.
Ahora hicimos cambio de pilotos. Ahora se une Joel y Chris. Vamos a explorar un poco más el área antes de empezar el descenso. Y vamos a... Can we pull up the pH on the screen if possible? Here, just Schmidt have it.
So was that?
We get pH
The pH.
on the screen.
Yep. How was the work on? Nice for the play.
7.85. Nice. Ideally, we'll head up canyon a little bit towards this top of the slope if possible.
Map out so I can see where the stick is. Nice, it's straight ahead.
Yep.
Alright.
Perfect. And we'll see what we can see.
Oh. it's connected to something, so this was not it. Let's do this. We don't need this waypoint one, do we?
What?
Do we need this waypoint line? No, we don't.
The waypoint, I think we
it's-
had one already.
Yeah.
We dropped one waypoint at the coordinates of the site.
Yeah.
So I think we should be good because we came back and
But on the north side of the line, if you take it a left.
One straight. One straight in front, straight ahead, right? So just go there.
seguimos en esta
Opposite direction.
tarde estrellada con estos ophurios, estas estrellitas de mar.
Right? Zoom out. Zoom out.
A 918
No, we're gonna go the other way.
metros de profundidad.
Okay, so is that correct? We're going the other way.
Think about direction.
According to the map. Oh, if you're gonna cross this over the other side.
You can look at it here, Sylvia. So we are here, right?
We're trying to go up the slope.
Yeah, so then we go to the northeast.
But we run northeast.
Or it's a bit.
Yeah.
Alright, so yeah. No, I don't want him to go up the slope. We need him behind him. He needs to turn his heading and come around the other side. We don't want this. We don't want the ship. Yeah, we want the ship in shallower water than us because that makes it fun.
So I'm gonna move it.
So
You're gonna follow this line or? The line? Are you following this line?
yeah, I'm going to go eventually. So we need to get him to. Yeah, but we need him to turn his heading so I can come alongside of him so that I will be in shallower water than him.
Great, so we.
Yeah, great.
Okay, so the next move is gonna be towards the northeast. And for that, we need you to change your heading so you are behind us or to the east of ROV. So I gave you a waypoint right now. You can start heading there and changing your heading. Is that possible?
Yeah, so which heading would you like us on?
200.
200 would be fine.
200 around? 200?
200, okay.
Thank you.
So, in this situation, this is great.
Somehow made a mark.
Yeah. The thing is hit it, and then right click on it, and don't go to it.
You happy with the cameras?
Yeah, we do all of it.
seguimos explorando un poco esta área. Ya pudimos obtener los sedimentos y bueno, vemos la cantidad de materia orgánica que está siendo depositada es muy evidente. Es un ambiente en donde en cada una de nuestras transmisiones hemos estado viendo esta nieve marina, que es materia orgánica que cae, son detritos de descomposición que viene desde la superficie y cae en los sedimentos, y esta materia orgánica termina de descomponerse, termina de reciclarse en estos sedimentos. Las capas de las que mencionaba Silvia, los procesos de los que ella está interesada son procesos anaeróbicos, son procesos que se llevan a cabo en ausencia de oxígeno, y estos procesos son muy importantes ya que permiten reciclar toda esta materia orgánica que se deposita. Si no existieran estos microorganismos y estos procesos, habría una acumulación muy grande de materia orgánica. Es como si no se recolectara la basura del planeta por años, eso completamente cambiaría, digamos, pensándolo en los océanos, la dinámica de los océanos. Si la materia orgánica no se reciclara y no se convirtiera en otras fuentes de nutrientes para los seres que viven en el océano, la química o el engranaje de estos procesos se perdería si no existieran estos microorganismos anaeróbicos. Can you zoom into? Oh, it's just a crab making a little bath. Oh, he's not very happy. He's not very happy.
We will find a bacteria that hasn't been described before. We'll probably find something, just because there has been not a lot of research done on the community composition here. So that's going to be amazing, whether it's exactly what we're looking for or something completely different. We will find something new.
RV Bridge, you let me know if you need me to go any faster. I'm just dawdling here.
Your speed is just fine now. Thank you.
Okay.
cuarto acaba de entrar también Emily Hyde, que es estudiante de doctorado de la Universidad de Texas en Austin. And she's been working with water and working with sediments. And so one of the questions, Emily, in the chat was about marine snow. And what can you tell us about the productivity in this area? What can you tell us about the high productivity in this area in terms of organic matter and the marine snow that we're seeing here?
Yeah, so
Wait, it's not. I don't think it's on.
There's something with that mic.
You want to come here?
Hi everyone, sorry we had some mic problems. So in this area we have a high amount of organic matter. So this marine snow that you're seeing is actually really fueling the seafloor, not only in microbial communities, but also the organisms that we're actually able to see out here, like some of these fish and the crabs and some of the larger organisms that you see on the screen. So with this high amount of organic matter that's raining from the upper layers of the ocean, we can see that this is a fairly productive area. And a lot of this that we're seeing here is basically just brought from the upper water columns, but we also are still fairly close to the mouth of the Rio de la Plata. And so we could also still be getting a little bit of flow from that area as well. So this is a very productive area. And we can also see that because we're pretty close to a sampling site that we were at last night where we had really organic matter rich sediments, and you could actually see that whenever we brought those up on board, and they have quite a smell to them, high in sulfides, so they're very, very productive. But these sediments that we brought up last night were anoxic, so a lot of the layers don't actually have any oxygen present.
Ahora vamos a cambiar el turno del watch lead. Professor Boss and Daniel are gonna. And Emily, you're good. Okay. I thought it was Daniel. I'm sorry. We're gonna. Whoa, what was that? Going to. To change the watch lead. Y bueno, me despido yo también. Gracias a todos los que siguen sintonizando. Quédense más para tener estas imágenes. Vamos a continuar un poco más el live stream para explorar esta área. Y nos tenemos que preparar para cuando suba el submarino poder procesar estas muestras.
Thank you, everyone. I'll be handing it off to Buzz here in a bit.
Y en esta expedición, simplemente para todos los que nos escuchan, esta expedición es internacional. Habemos científicos de diferentes nacionalidades, de diferentes universidades. Mayoritariamente de diferentes partes del mundo. Yo soy mexicana, pero soy científica en la Universidad de Viena. Professor Baum es en Reino Unido y les va a comentar en inglés, y Emily Hyde también. Así que hagan sus preguntas. Y nos vemos en el siguiente live stream. Un saludo muy grande a Viena, con mucho cariño, y hasta la próxima.
Hello. Yeah. Good afternoon, good evening, good morning, wherever you are in the world. This is the live stream from Falkor 2, searching for the Asgards as part of the Schmidt Ocean Institute.
Hour? Yep. To come up? Yep.
We're sampling
It's 6:15.
the bottom
So you come up at
of the sea off the coast of Uruguay. And in this dive, we've collected the cores that we need, and we're just exploring, chasing brittle stars across the seafloor. And I'm joined by Emily Hyde.
Yes, hi, Buzz. Yes, I'm Emily Hyde. I'm a grad student in
Emily's going to just join us in a second. He's a grad student in Brett Baker's lab and spends a lot of her day plugged into a water with purifier to filter water from different depths in the ocean, and then to sample the things that are caught in the filter for their DNA to find out which microbes are present at different depths.
Yeah, so I only caught the last part of that, but from what I heard from Buzz, just a quick introduction. So yes, I am doing a lot of the water filtering. So not only do we have the wonderful Sebastian that we're able to go to far depths and able to take these push cores so that we can learn about the microbes that live in the sediment, but we also have a CTD on board, that stands for conductivity, temperature, and depth. And what that is it's looking at the water column. So it's taking Niskin bottles and water samples throughout the water column. So not only do we get to learn about the microbes that are living in the sediment, but also about those that are lurking in the water column. Most of the time on board, you can find me in the hangar where I'm filtering liters and liters of water to look for the microbes that are in that column.
You're looking for what you would like to find?
Yeah, definitely. So we are looking for Asgard Archaea while we are on board. So Asgard are the closest microbial ancestor to eukaryotes. So complex cells, so all of these fish that we see on the screen, and us ourselves. So we're looking for more lineages of Asgard Archaea, not only more lineages, but just more genomes as well. So specifically for the work that I do in Dr. Brett Baker's lab, we are looking for sequences. So we are going to be doing metagenomics and metatranscriptomics. So while there's lots of people on board who are looking to culture some of these organisms, we are looking to get their genomic information so that we can learn more specifically phylogenetically about these organisms. So we are on the hunt for Asgards, whether they are aerobic or anaerobic or somewhere in between.
And Emily, can I ask you, how will you know if you've got an Asgard, and especially if it's a new one? How would you identify it?
Yeah, so the way that we would, so on board, we are using a sequencer while we're here, and so we're actually able to just do a quick identification from specific sites. But when we get back home, we're able to do a more in-depth analysis. And so what we'll do is we will sequence the DNA, and once we have that DNA sequenced, then we're able to build genomes from this basically raw metagenomic DNA. And so once we have those genomes, then we can basically analyze those further to place them on the tree of life, and that will allow us to understand more about these about Asgard Archaea and where they fall in the tree of life, and if we have any more lineages specifically.
And the way you do that, I guess, is you have a sequence of letters, ATGC, from a sequence, and then you have to compare it to all known sequences. Is that right?
Exactly right. Yeah. So, the raw data quite literally just looks like A, T, C's, and G's, just like you learned in early bio. And from those A, T, C's, and G's, we can figure out what proteins these organisms are actually able to make. And we use what's called a set of marker genes. And so these marker genes are conserved across the archaeal tree of life, and we also look at ones that are conserved across the entire tree of life. And using these marker genes, we can actually place them phylogenetically to understand where they fall in the history of life. So it's these marker genes that are really important to us. So that's also something that we can do a quick search for while on board or while we're back in before we have full genomic data. We can actually do this before we build the genomes. We can do this just from the metagenomic data itself.
Cool.
Buzz, I'd love to hear a little bit more about what you're able to do on board. I know we do very different things while we're here on board. So if you could tell us a little bit more about what you and Fraser are able to accomplish while on board the Falkor, too.
Thanks, Emily. I mean, essentially, I'm Fraser's assistant, so Fraser works in my lab, but I do most of the writing, sitting in my office, and Fraser is an experimentalist who has mastered the art of culturing Asgard Archaea, which is a rare skill in the world. There are only a few people who can do it. And so when we collect sediment samples, which are quite rich in microbial communities, what we're going to do is take some of that material and take it back to the MRC LMB in Cambridge, where I have my lab, where there's a team, in addition to Fraser, who work on Asgard Archaea, and we'll try to we put a small amount of the sediment into a bottle, and the bottle is anaerobic, so we try to chemically remove oxygen, and we grow it then in we flush out the oxygen as well as getting rid of the oxygen, and grow them in anaerobic conditions. And the hope is that some organisms in different bottles will grow up, and that from an enormously complex community like a rainforest, a microbial rainforest, out of them over time will grow specific lineages that take over, and in the end you have a bottle that's dominated by a small number of organisms. And we hope that it includes Asgard Archaea. And one of the special things about Asgard Archaea, as well as being related to our ancestors, the cells that with the acquisition of bacteria became complex cells, Asgard Archaea also require partners to live with. So they can't, it seems they can't live alone, either by parasitizing or sharing with other organisms, microbes. They're able to live. And so within each bottle you actually have a community, hopefully a simple community, where you have an Asgard and maybe some partners. And then once we've established that, which the last cultures took Fraser 6 years, so we just put a paper online reporting our findings, took 6 years. But in that community, because Asgard are stable and dominant, we can actually start to do experiments to see what do they do, how do they look, what's their structure, what's their lifestyle, what's their behavior. And so and in the samples Fraser's analyzed in the lab that he has been able to culture, we've seen amazing things that break the paradigm of once upon a time there were simple cells, bacteria and archaea, and then complex cells arose 2 billion years ago. What we've discovered is that Asgard Archaea are a sort of bridge between the simple and complex. They're archaeal cells, which tend to have small genomes, simple, they look genomically simple, but they appear to have some of the complexity that we thought was unique to complex cells, that, like in us, in fish, which I'm looking at now, in brittle stars, in squid, in plants, in fungi. So that's the kind of thing we're essentially hoping to be able to cultivate sort of new species, which we can then study in detail in the lab. But it's a long project that begins with taking a push core from the bottom of the sea, and then maybe in years to come we're able to see the creatures that we've taken with us.
Thank you so much. That was such a good explanation. I do wonder if anybody listening online is also curious, why does it take so long to grow these Asgard Archaea? What is unique about their life cycle that makes it, you said Fraser grew these for 6 years. So can you just tell us a little bit more about, what is unique about Asgards, or not unique, that is interesting in that sense, where what takes these cultures so long to grow up with this dominant, the dominant organism being Asgard, or have enough of Asgards in order to study them and get them to a stable culture?
Yeah, I mean, that's a great question. And in a way, a deep question, because I think what many biologists actually don't appreciate is that when you learn molecular cell biology or genetics, you tend to work with a small number of model systems, we call them, which are actually the tools through which, the lens through which we have learned about life on Earth. So such as yeast that you use to make beer and bread, these little worms called C. elegans worms, the fruit fly, and mice, zebrafish, and E. coli, these bacteria from Asgards. And through a small number of these organisms, we've learned a lot about the fundamentals of how life works on Earth. Because all organisms are related, we can take that knowledge from one cell, say a bacterial cell from our guts, and apply it to understand better the cells in an octopus, how they work, because there are some things that are shared. But these organisms have not been chosen by chance. The reason that over the last few centuries people have worked on these so-called model systems to study the fundamentals of life is because these organisms grow easily in the lab, which means you can put them like yeast into a sugar solution with a bit of hops and you have beer, or you can grow the cells and study them. And so these are the kind of organisms that we study which grow alone, fast and easily. And so these are lab organisms. But what I've really only come to appreciate recently, and the reason I'm so excited to be on this expedition, is that that is a very small window into life, and actually other organisms live in completely different ways. And in fact, many organisms live slow. They don't boom and bust. They don't grow as fast as possible and then take over, and then we see societies try to do, like our picture of capitalism, survival of the fittest, you go fast, quick, furious. This picture of biology as a battle of the survival of the fittest and the fastest is not a good representation of much of life on Earth. In fact, there are many places on Earth, like in the sediments, where the community is, there's no space. So to live, better live slow and maintain a niche and survive there. And for some reason that we don't understand, and I don't understand, but I think in general it's not understood, is why some organisms have this different lifestyle, that they grow slow in communities and a sort of steady state, persisting for years, maybe hundreds of years, maybe thousands of years, while in other parts of the world you can see organisms that boom and bust quickly, taking over as much as possible and then running out of food and dying off. And one puzzle is, where is that written in the genome of an archaeal cell? We don't know that, but it's interesting because it seems to be a feature of many archaea. So, for example, when we take a core of sediments, if you don't add chemicals, antibiotics to kill the bacteria will tend to take over and bloom at the expense of the archaea, and particularly Asgard Archaea, that live slow and with partners. And so we have to learn to then study these other kinds of organisms that maybe are persisters, that live in this other kind of way. But it's challenging because they're not so easily tamed for the lab, which is partly why they're interesting. So maybe that helps to explain why it takes 6 years, because we take some sediment and you have to wait sort of 6 months for that to grow up to fill the bottle, and then you take a small amount of that, put into a fresh bottle, again removing all the oxygen, and it will again take a few months. And over a series of passages, over many months, the months add up to years, but in the end we can end up with a simplified community where the cells still thrive, growing slow together, but in sufficient numbers that we can study them. Was that clear?
I thought it was very clear. Thank you so much. I also just want to note, as you were talking, you were mentioning how, these are slow growing organisms and that, they require, maybe other organisms in order to partner with. And that's that's one of my favorite things about microbes, just in general, not only specifically Asgards, but we have, in the microbial world, both in bacteria and archaea, we see what's called syntrophy, and it's this close relationship that a lot of organisms have. A lot of microorganisms have, where they're basically passing back these substrates that are, their byproduct, and they're basically living together in really close partnership. It's a type of symbiosis, and it's to me, one of the most intriguing things about the microbial community, because, like Buzz said, this isn't something we can just learn from a genome, and one of the reasons why I think it's so great that we have so many different type of scientists on here who are doing different type of work. We have people who are culturing. We have scientists who are looking at the geochemistry. We have scientists who are just looking at the genomic data, and all different parts of the water column and of the sediment. And so when we pair all these together, we can learn a lot more about the entire lifestyle of these organisms that we wouldn't be able to tell strictly from a culture or strictly from the genomic data. And so being able to, slow grow these organisms in the lab and then coupling that with the genomic study really allows us to learn so much more about these Asgard Archaea. All right, let me go ahead and do a quick log really fast of these amazing brittle stars that are crawling across the screen. There we go. Oh, sorry, what? Okay, perfect, thank you.
Why did you do that? Alright, so yeah, let's do that.
All right. Are we?
I was trying to zoom in on that, but it just zoomed something else. So zoom in, discussion. So yeah. No, I don't know what we're doing. I'll find another one. Then it'll be right there. Zoom in on that.
I see a comment in the chat that says Asgard sound like hard work, and while I think a lot of the work that is not only being done on board but post this cruise is hard work, we're all very excited to get closer to understanding more about the origin of eukaryotes, which is what studying Asgards can tell us. So while growing up a culture for many years sounds like a daunting task, it's also one of the most exciting things that we get to do as scientists, is grow the organisms that we're interested in, or basically create a new genome from metagenomic data of the organisms that we're interested in, because then we get to understand more about our organism of choice. And for all of us on board, our organism is Asgards, which is a large group of organisms. It's not a singular one, but this phyla is particularly exciting to all of us because we're all interested in eukaryogenesis.
I mean, in some sense, also the fact that it's hard to grow is the reason we don't know about it. So often, if you're going into the unknown, you need to use a new tool, or you have to look at things in a new way. And Asgard were really only discovered through the technology that Emily's using, like doing genome sequencing of environmental samples. So this has only been possible in like the last 10 years or so, to go into an environment, collect DNA and sequence it, and then assemble those small reads into genomes, which can then tell you which organisms live there. And that revealed things so the new technology of genome sequencing revealed this whole new, opened up a new vista in a way to see life in all its diversity that we'd missed. And I suppose since then the hunt for Asgards has been on, and we've been on it, and then the subbiology has been hard, but many people around the world now are now culturing them, and we're starting to open up this window into these amazing cells. So it's hard, but that's also the reason it's not known. And of course, as scientists, we like to be on the cusp between the known and the unknown, searching for new principles, new organisms, new ways of being in living systems.
Yeah, and I also want to note, this, for those listening, the world of Asgards is still fairly new in terms of, the first Asgard genome was acquired, just as Buzz stated, through metagenomic sequencing, and this was at Loki's Castle, which is a hydrothermal vent field in the North Atlantic. And from this area, they were able to get a genome of the first Asgard, which is Loki Archaea. And so that's actually where, the name came from, Loki's Castle, Loki Archaea, and then that became the phyla Asgards. And so this is still a relatively new field, which means that, being able to sequence new genomes from new areas means that we could find completely new lineages, which is one of the other very exciting things, is that, we're all excited to be on the cusp of this, all of these new discoveries, because this is still a young field. And, we're still looking at different methodologies to study them, because just as Buzz said, there's a lot of studies done on model organisms, and Asgard is not a model organism. Model organisms, as he stated, tend to be fast-growing and non-dependent on another organism, which is neither for Asgards.
Yeah, so for people who are just joining us, we're sitting in Sebastian. Well, we're attached to Sebastian, the ROV that's at the bottom of the sea off the coast of Uruguay, several hundred meters down, 885 meters down. The water is cold, but there's actually quite a lot of oxygen, and we're just tracking the bottom, looking at the wonders that the cameras on Sebastian can reveal about life in this part of the planet. And apologies for not speaking Spanish. Some of the scientists on board are locals. We have a whole team from Uruguay, which we're thrilled to have as part of the group, because they know these waters and they're also experts. And also we have many Spanish speakers who you'll be able to hear on other live streams. But for now, it's just myself, Buzz Baum, from the MRC LMB in Cambridge, and Emily
from UT Austin. And I'm part of Brett Baker's team, who is the chief scientist on this expedition. I do see a question in the chat that is asking about the ecology of Asgard Archaea. And so for the most part, we find, well, actually kind of two-part. So the first thing I'll note is that in an amazing way, Asgards are found nearly everywhere that we have sequenced, but they're not found in a high abundance in a lot of areas, which is one of the reasons that we are interested in studying this area, because this has a high potential for there to be a high amount of Asgards and the high relative abundance of these organisms in this area. And that's due to the fact that we have more recently learned that Asgards tend to dominate in comparison to other areas, dominate in terms of the archaeal abundance in coastal marine sediments. So we are off the coast of Uruguay, and particularly in oxic sediments that have an oxic layer still and are not fully anoxic, because there are Asgards that have the potential to utilize oxygen, and there are fully anoxic Asgards as well, or anaerobic Asgards as well. So we can see that there's both ecologies in terms of their dominant electron acceptor and basically what they're able to live on. And
can I tell something? Can you just tell us how do you deduce that? Because as I understand it, you're just getting genome sequence, and from that you're figuring out the the sensitivity and ability to use oxygen. Is that right?
Yeah, exactly. So basically, the way that studies have shown that these Asgards are capable of utilizing oxygen is, once we have a genome, we're actually able to predict the proteins. Oh. Okay. We're actually able to predict the proteins. Let me go ahead and record that really quick. There we go. Okay, so we're actually able to predict the proteins, which allows us to determine the metabolic potential of these organisms. So while we can't conclude that until we're actually able to look at, we can look at a living organism in a culture, or we can also look at the transcriptomic data, so we can actually see what pathways these organisms might be using. But from the genomic data, we can infer that some lineages of Asgard Archaea are actually able to use oxygen. So basically they have these genes that have the potential to utilize oxygen, and they also have genes that could be reactive to oxygen and scavenge it. So there is this potential for organisms and Asgard organisms to actually be able to live in oxic areas. But the majority of Asgards cultured so far are actually anaerobic. And so we are also interested in searching for these, which is why we're looking at the entire column, not only of water column, but also of the sediment, because there will be in most cores that we take from this area, there's going to be a layer where it basically switches from oxic to anoxic sediment. So not only are we interested in that oxic area to look a little bit further into these Asgards that can utilize oxygen or have the potential to utilize oxygen, we also are interested in looking at these organisms in the Asgard phyla that are living in the anoxic area, that are not able to utilize oxygen, or that we don't currently know can utilize oxygen. And for example, the Archaea, which was the first Asgard to be studied, these cultures are anoxic. So there's a lot of cultures of these not a lot relatively, but we have more cultures of Archaea. And it's exciting because you can also see that these are anaerobic, but we're also on the hunt for more aerobic Asgard Archaea on this specific expedition.
And one of the reasons I think this is an exciting avenue of research, looking at the ability to use oxygen as an electron acceptor, and also oxygen toxicity, is because 2.5 billion years ago, when Earth was populated by bacteria and archaea, as we understand it, there was very little oxygen. And then around 2.5 to 2 billion years ago, there was a rise in oxygen across the planet, which gave rise to oxygen in the atmosphere. And this killed probably a lot of organisms, because oxygen is actually a very toxic molecule. Wow, beautiful ray on the live stream. Gorgeous, small, beautiful. So not only is it toxic, but if you can use it, you can do amazing things. As we know, when we burn fossil fuels, for example, to power a jet plane, we're using oxygen. So this led to the idea that now we know that complex cells arose from the coming together of archaea, which tend to be anoxic, as Emily described, these Asgard Archaea, and bacteria, which often are a bit more oxygen tolerant, and potentially some of which could have been early users of oxygen as electron acceptor to generate energy. It's possible that this coming together of these two organisms, a bacterial cell and archaeal cell, could have enabled archaea to leave the oxygen poor environment and to make their way into oxygen. And all the eukaryotes you can see in these beautiful images from Sebastian, this gorgeous ray, and all the brittle stars and everything that's big you see on a walk in the forest, all these organisms which are complex cells, they all almost all of them need oxygen to live. And so somehow during this history of from simple to complex cells, oxygen played a key role. And so if we can identify Asgard Archaea that are oxygen tolerant, or maybe even oxygen users, then it might help us understand the transition. Of course, it's also possible that many of these Asgard Archaea, of course, they are not our ancestors, they're related to our ancestors. So it might be that in the 2 billion years since, they've learned to use oxygen as we've learned to use oxygen. But if you can reconstruct the lineages, we might find that some capacity to use oxygen might have pre-existed before the emergence of complex cells such as us. So for this reason, all these studies into Asgard Archaea and their ability to live in communities and to use oxygen are really interesting and may promise to shed light on this major transition in the history of life on Earth, where from a world covered with bacteria and archaea, simple cells, structurally simple cells, emerged these wondrous creatures, brittle stars, fish, everything we can see that's large in the ocean, eukaryotes. And actually, one thing for me was very exciting today, actually, is that earlier on we came across a reef 200 meters below the surface, so quite a deep coral reef, where there were bacteria that were forming multicellular colonies. So it's not just complex cells, eukaryotes, that can form complicated multicellular structures, but also bacteria. And I had never seen them before, but Emily, can you tell us a bit about that?
Yeah, so earlier today, we have another wonderful king crab.
With a fish.
Yes. It looks like
Can we zoom in on the...
Well, we don't have a regular camera, so
Oh.
using it for me. Don't be shy now.
Is it symbiosis or is it... This is always the question, right, Emily, with you see organisms together and you don't know if they're like the barnacles and the crab,
Yeah.
are they friends or foe or... That's a beautiful picture.
This is amazing. Oh wow, and we also have some anemones on
Oh yeah, maybe it's
in the crab.
Wow, it's
Maybe
a walking...
parasitic.
It's like a walking community.
Yeah.
Yeah, crab's got a couple anemones, bunch
Yeah.
of gooseneck barnacles, and then you got that eel pout hanging out with them.
Alex, you think he's okay, or the crab?
We've seen a lot of them with the barnacles around. I mean, I'm sure it's preferred to not be covered in barnacles, but we've certainly seen a fair share of them cruising around with a decent amount of barnacles on them. There are parasitic barnacles that target crustaceans, especially like there's been studies up in the Alaska crab fisheries and that sort of thing. But those barnacles tend to be underneath the crab. Yeah, so I don't really know the relationship here other than it provides some sort of substrate for that gooseneck barnacle to attach to, it looks like.
Yeah, I mean, you could say that studying Asgard Archaea is a strange thing to do because they're these peculiar small organisms, but barnacles too look maybe uninteresting, but Darwin spent many, many years of his life at the microscope with barnacles, and I think it distracted him so much that he almost failed to finish. Is that true? Origin of the Species. So, biologists, we often look for clues in the small. In fact, you can see the barnacles are amazing.
Yeah.
Little fans, feet.
Very cool.
Wow.
Here we are without our regular science camera right now. We don't have a regular science camera.
Amazing.
So it's a little cool.
Great camera work. Really, really good. Amazing.
Especially since we're one camera down, right?
Yeah.
That was so incredible.
Yeah, for anyone joining late and you're like, what is this view? Our science camera is down at the moment. So we're going to take a look at that once it's back up on deck. But the pilots are doing a fantastic job of sort of lining good shots up with the situation cam, which is what you're looking at now. And that is able to tilt and zoom, but it can't pan. So you got to line things up manually with the ROV, and then you can zoom in.
Buzz, I'm sure that you know, but
Well, tell me.
Sorry. No, this is actually not a science-related question. Just making sure. Do we know when we're supposed to head back up?
In 11 minutes.
Oh, sorry, what?
11 minutes.
11 minutes. Okay, great.
Yeah.
Cool. I just want to make sure that I didn't need to do anything. Okay, perfect. So, Buzz, before we saw that amazing crab and all of the barnacles, you had asked me a question, and I'm trying to get back to that.
Oh, yes, it was
trying to
about
remember.
the multicellular bacteria.
Oh, yes. Okay, so earlier today, if you were also on the stream, we visited a amazing reef that was just about 200 meters below sea level. We are quite far from that, or not too far from it now, but we're 900 meters down. We're 871 right now, so we're at a very different area than we were earlier. But when we were there, we were actually able to see some microbial mats, just off of sight. I can't, positively identify what those mats were, but they were likely some sort of Geochemistry or Thiamarita or Thioploca, and those are just basically types of filamentous sulfur oxidizing bacteria, and they form basically a type of microbial mat that you can actually see with the naked eye. And what was really interesting about those mats that we were seeing earlier is that typically, these mats do come in different colors, but because of the sulfur oxidation, a lot of them are actually these like white, they almost look like streamers. It's like white streamers that are basically attached to something, whether it's the reef or a rock. I've seen them attached we have a little jelly down at the bottom, just a quick spot. So, I've seen them attached to, more shallow, they're in freshwater as well, or at least the Thioplocas are. And so we saw these earlier today, but there was a few that were this like really light blue color.
Like a veil, almost like veils as well.
Yeah, it was really interesting. And that was actually something that has been in conversation with multiple of the science crew on board today. I've been chatting with different scientists, us coming up with ideas of what it might be. There's some pretty obvious possibilities, but then there's also some, more far-fetched that we're like, oh, but what if it had been this? So it's pretty cool to get to see something like that, because I have done a lot of work, not within the sulfur mat world, but within the sulfur world, and I've never seen that blue tint on any sulfur oxidizing mats like that before. So it was really cool to get to see that on Sebastian today. So that is just an example of one of the microbes that are living in these complex communities that we can actually see with the naked eye. You can't see the individual cells, but you can see the community itself that is working all together to basically oxidize the sulfur, which means that area had a high amount of sulfur to begin with. And so that's what all those organisms are living on. And a lot of these sulfur oxidizing bacteria are going to have some really close and really interesting relationships. For example, we see them at hydrothermal vents, and they have close relationships with the worms that are living at hydrothermal vents.
And I suppose one question one could ask is what is it about? Because it tends to be true that complex cells, eukaryotes, form redwoods, sequoias, blue whales, squid, these huge multicellular things that we can see, including ourselves, of course. Whereas bacteria and archaea they live in communities and can form a community consortium structure, but less often see them amongst themselves forming complex multicellular objects. And I think, I mean, maybe Emily knows about this a bit, but and she could maybe say more than me, but one of the reasons is that they tend to have small genomes, which doesn't give them much to play with. So they, I think we're heading up soon. So we'll say farewell to the bottom soon. They tend to have smaller genomes, which means there's less flexibility to play with. Each cell essentially is doing what it can to metabolize its food, live in the environment it does. In addition, partly because of that, they can't assume many states. So they can have states of physiology, so when there's high oxygen or low oxygen, you might change a bit. But one thing that's special about complex cells is there's lots of extra information that they can use to do different things with the same genome. So an organism which has the same DNA a cell with that same DNA can choose to be a sperm cell, an egg cell, a muscle cell, a neuron. They can differentiate, the word is, to become different kinds of object. And so together, these different cells, guided by the same genome, can evolve to do quite amazing things, like swim if it's a fish. And bacteria and archaea don't seem to do that nearly as commonly. There are exceptions. Cyanobacteria can form chains in which some cells take on a different state to other cells. But typically, a community of bacteria and archaea, the bacteria and archaea will be doing a similar thing if they're in the same conditions. And so it's a different kind of community to the one that you can see inside a single fish, where there's a community of cells, a bit like a biofilm, except it's a biofilm where everybody's invested in the same thing. They're all contributing to fishiness, and a subset of cells are destined to give rise to the next generation, while the rest are going to die.
Yeah, the complexity of microorganisms, or the lack thereof, compared to eukaryotes is one of the most interesting things, because they basically have to, in very simple terms, they basically have to get creative in how to live their life with these really small genomes, and so they're able to form these really complex relationships with other microbes in order to get all of the things that they need. Because, like Buzz said, small genomes means that they might not have all of the, for example, metabolic pathways that they need in order to create the things that they need to function. So, for example, we have a lot of microbes that are chemolithotrophic. So that means that they are going to have to basically, in simple terms, make their own food. They're going to have to make their own food from, not from, it's not photosynthesis, it's chemosynthesis. So they're actually making it from the minerals that are around them, basically from these gradients, these chemical gradients that they have near them. And so because of this, a lot of the time, not only are these organisms, having to form these really complex relationships, but they're living in these communities in very specific gradations. For example, in a sediment core, you'll see really distinct microbial communities across that core based on the geochemical gradients across that core, which is why it's so important for us to have this variety of scientists on board, including those who are doing geochemistry, because the geochemistry itself of the core or of the water column can actually give us a lot of ideas about who might be living there before we even sequence the data, before we even culture any of the organisms that are there. It can kind of give us a nice predictor for who we might expect to be living in that area, because we have an idea of how these organisms are working together. But what's so cool is that there's so many microorganisms that are unknown and that we could find while we're out here. And there's also just a lot of metabolism, parts of metabolism across the microbial tree of life that we just don't quite understand fully yet, which again is to me one of the most exciting things about studying microbiology.
Yeah, that should be okay.
Oh, looks like we might be
We'll do that.
headed up.
Yeah, I
Okay,
think
awesome.
we're
Yeah, wait
saying
till you get right inside the
farewell
bus before bringing the head in my morning.
soon to the bottom.
Yeah, copy.
Because we've already collected earlier on this afternoon some sediment cores, and so they are in a rack on Sebastian, and once Sebastian gets to the surface, those are the precious tubes of dirt that we're going to work on for the next many years.
And Buzz, do you happen to have the log up there to see if there were any Niskins that were fired?
I don't see
Let me bring the notes to you.
Oh, it's totally fine. It's just wanting to see. Let me go ahead and I'll come over. These two Niskins were fired. Okay. And they only wanted. Do we not have a full porch, of course? For this one? It's special.
Do we know which cores we have? Do we have a full?
We didn't do any of the course.
Oh yeah, you guys things.
I know that they did. They were working on the porch. We don't have.
Oh, I see. There's the numbers have got switched. So 6.
Yeah.
Okay.
Put them in different spots than what they were originally. So the numbers so it's wrong, but
Toilet, Roger.
Do you guys want to do it at the same time?
Oh, yeah, sounds good.
Yep, so we're heading up. There's 850 meters above us of water, and on the way up, well, let's see what we can see, but for the moment it's warp drive through the snowstorm. One thing you said, Emily, that I sort of often reflect on is that we talk about that if you live in a microbial community, some cells can make some things, other cells can use them and in return make something else, which they then can, as a community, live together and in that environment, that whereas by themselves they'd find it hard. And I think that sometimes when you have a group of scientists coming together, it's similar that together, all our different expertise, we can use that diversity of skills to also study these organisms and their communities and their diversity of skills.
He's going to try to make a meal out of that. And then he didn't do anything. We were like, come on, dude.
Should we go ahead? Okay. So we are going to go ahead and wrap. Ooh, we actually have an amazing jelly right as I was about to wrap this up. Let me go ahead and log this incredible jelly.
I think it's Tina 4.
Oh, Tina 4. Okay, perfect.
Gorgeous. Wow. Look at
Wow. Oh my gosh.
Oh.
Oh.
you can see that this is not a community. This is an individual with a singular identity as a whole, in which the cells are all acting, to further the behavior of the organism of which they're a part. Beautiful.
Incredible. But I do know that we're just given signal that we should go ahead and sign off. So I just want to say thank y'all all for listening and streaming tonight. I was so happy to get to chat with Dr. Buzz Baum today about Asgard Archaea and all of the incredible science that is happening on board from this wonderful science team, as we have geochemists and microbiologists who do all different facets of the ecology and evolution of Asgard Archaea. So it has been wonderful to get to see the end of this sampling, and I'll leave it over to Buzz to give the official sign-off as the watch lead.
Well, thanks, Emily. Yeah, it's been a pleasure co-hosting with you, and also we should thank our amazing Schmidt Ocean Institute team, the crew who've done an amazing job without the science camera, still showing us dazzling pictures of the world at the bottom of the sea. And so yeah, please join us again with the rest of the team, where there'll be more Spanish speaking and also more science, more beautiful images from the Searching for the Asgard Spark team. Thank you. Goodbye.
See if you not put the science cam up on one of the